And welcome to the second summary video for the GCSE topic of cell biology. In this lesson, we are going to be looking at microscopes. We want to explore the different types of microscopes and introduce new vocabulary in which is focused around microscopes. Therefore, our key focuses for this lesson are the vocabulary itself, in which we use to describe microscopes in science, as well as the differences between light microscopes and electron microscopes. So to start, I want you to take a moment to look at the important terms. When we describe microscopes, we have two different lenses. Firstly, we have our objective lens. This is where the lens meets the slide and can be changed between different magnifications. Your typical light microscope will have a 4 times, 10 times, and 100 times magnification on the objective lens. The second lens is the eyepiece, in which, of course, you'll be looking through. We also have the definition for magnification, how much larger the image is compared to the real object, or how much more magnified, zoomed in, that image is. We actually calculate this by multiplying the objective lens by the eyepiece lens. Lastly, we have resolution. And resolution is just how clear the image is. Normally, when we introduce the term resolution, there's a lot of confusion about what resolution actually means compared to magnification. We hear resolution very often when we talk about things like our TV screens or smartphone screens, and a high resolution we just associate with being good. The thing about resolution is it describes the clarity of the image. Magnification is simply how enlarged or magnified the image actually is, how zoomed in it might seem. And a lot of the times, magnification and resolution actually do not come hand in hand. The more magnified an image is, typically the resolution goes down. In this row of images below, we can see the face of a dog. You have what is described as high resolution. It's very clear. You can see a lot of the details, the spots, even the grain on the wood. Whereas low resolution, you can barely make out the dog's face. With magnification, we zoom in and we can see a lot of the details a lot larger. So the eyes of the dog look bigger. However, it doesn't make the grain of the wood in the background any more clear, or the hair or fur on the dog's head. In fact, as we magnify further, we can actually tell that the image is of a lower quality. It just simply looks a lot blurrier, it looks a lot less sharp, and you can notice the difference in resolution. Moving on, looking at the diagram here of a light microscope, we can see all the labels clearly posted for the different parts of our light microscope. We've already talked about the eyepiece and the objective. The tube is just used to connect the two lenses together. It is usually a series of mirrors in which reflect the image up into the eyepiece from the objective lens. The coarse focus and the fine focus knobs adjust the stage as well as the tube accordingly to get a higher resolution image. You have your light source on the bottom. With a light microscope, of course, it is usually going to be electric and therefore just shine a small incandescent light into the slide. And lastly, you have the stage, which actually holds the slide piece in which your specimen will be on. On this slide, we have a bunch of examples for specimens that you typically see under a light microscope. In the top left here, we have what we call planaria, which are very small microscopic water organisms. You have an example of an onion cell on the top. On the far right is an example of red blood cells. And on the bottom, we have an example of another microscopic organism called a tardigrade. So let's shift our focus a little bit and move away from light microscopes. On this slide, we have an electron microscope. And electron microscopes work by using a high voltage to emit subatomic particles called electrons through a series of lenses and through the specimen plate to reflect back an image onto a camera. Basically, using electrons, they can tell if there's something there or not based on if they reflect back. And if something reflects, it can create an image, as we can see on the following slide. We have two different types of electron microscopes. We have the SEM, which stands for Scanning Electron Microscope, and we have our Transmission Electron Microscope, or TEM. And both have very different functions and uses, although they use very similar methods in which they gather their images. A scanning electron microscope scans the surface layer of the specimen plate. So in our two images, we have pollen grains as well as bacterium. And the scanning electron microscope gets a very cool, very pretty picture of what the surface of these particles and cells look like. So we use scanning electron microscopes to get just a nice, fine surface photo. The transmission electron microscope, rather than get the surface, it actually transmits the electrons just a little bit further through that first membrane. And the images, instead of being of the surface, are of the internal structures of our cells. So for the pollen grain, we can see the nucleus, we can see all of the organelles like the cytoplasm, the vacuole, as well as the cell wall. In our bacterium, we can clearly see that there is a flagellum and other internal structures. So it's very cool to see these incredibly zoomed in, magnified images. And electron microscopes can magnify up to 10 million times the actual image size. So it's very cool to see images that our naked eye would definitely miss. On this slide, we have more images taken from scanning electron microscopes of everyday objects. So even on snowflakes, we can see the crystallization and the fractal of water vapor as it forms into ice. Additionally, you have sodium chloride or salt particles, and even the face of a housefly. And you can just see all the little details in which we definitely won't be able to see with our eyes alone. So in summary, there are two types of microscopes: light microscopes and electron microscopes. For light microscopes, there are two lenses. One is called the eyepiece lens, in which we look through, and the other is the objective lens. Magnification, by definition, is how zoomed in the image is, while resolution is how clear the image is after being magnified. And lastly, electron microscopes can be divided into two categories: transmission electron microscopes or scanning electron microscopes. Each provide different types of images for different functions. So thanks for sticking around, and I'll see you in the next one.